Hi, everybody. Today's lecture is on Chapter 4, Sensation and Perception. This chapter will cover and we'll get into descriptions about what is sensation and perception and how they are distinct activities. We'll spend most of our time discussing the visual pathways or how we see the world. We'll also talk about recognizing things that we see, and we'll end the discussion with hearing, the body senses, and the chemical senses. So, sensation and perception are distinct activities. Sensation simply relies on our body's senses to break down information in the real world into units that our brains can ultimately assess, if we think of it this way. So, light as it exists in the real world has to be um, received by our eyes, and we have different receptors, photoreceptors in our eyes that are able to recognize various wavelengths. And through transduction, we're able to convert these signals, the photons in the real world, into neural impulses, where we can process those impulses to perceive or perception the things that are around us in our environments. So, sensation is a simple stimulation of an organ sense. It's really the sending of signals to our brain about the world that we're in. Whereas perception, it's our brain's ability to recognize, identify, to really interpret the signals of sensation to form some mental representation of the stimuli we've received. Transduction is a process where the senses we have convert the physical stimuli in our environments into neural impulses, signals, to the central nervous system or from it or with it, depending on which sense we're talking about. And each sensory receptor that you possess is uniquely sensitive to a particular type of energy. It's weird to think of perception um, as an illusion, but it really is when you think about it. The world only exists in your mind. So we perceive rich, detailed, and a complex world through a relatively small subset of sensory signals. It's kind of hard to imagine your world as an illusion, but when you look outside and see the objects in your environment that you don't have two black dots in your visual field, but the reality is the optic nerve exits your eye, and where it does, there are no receptors. What we have is a process of perceptual filling in. Effectively, our visual system creates an illusion by filling in the areas where the optic nerve leaves, that perception of the world is there. It's real. I mean, to a degree, it is, but that's the illusion part. It's that filling in of the world we receive. We also can experience processes called sensory adaptation. You've probably all seen similar diagrams to this, where you're asked to stare at a fixation point for an extended period of time and then to look away. And when you look at a wall, you'll see an image appear. That's really called an after effect. But in this case, the adaptation is where the sensitivity of these neurons firing to prolonged stimulation can lead to a decline over time. Effectively, you've adapted to it, even though the conditions really haven't changed. We have a stronger response to stimulation than to constant stimulation. So we see different patterns of firing as the stimuli in our environments change rather than just staring at the same thing over and over. So if you stare at that image on the screen right now, in particular the black dot, what you might notice after several seconds is that that gray cloud around the dot will eventually disappear. This phenomenon is called the troxler fading, is an example of adaptation, where you'll see that the cells, they're firing before you do this, and the rate of firing changes as you stare at this, like I said, for several seconds. Psychophysics is an important element in our understanding of how sensitive we are each sense, and in particular, we'll deal with vision up front, is to change. So psychophysicists often measure the minimum amount of a stimulus needed for detection. Historically, you can imagine this would be done by candlelight. How many candles would have to be present at a certain distance for you to detect the light? Now, it's not surprising how sensitive your eyes are to light. In fact, in the evening, I'm sure many times you've looked up and saw the stars. Think about how far away that light has traveled, how many light years to reach your eyes at that moment in time. Psychophysics employs methods that systematically relate the physical characteristics of a stimulus to your perception. Imagine you can detect light by candlelight off in the distance. How many more candles would have to be added for you to detect an increase in brightness? So, psychophysicists are really important in furthering our understanding of how sensitive each of the systems we possess, the sensory systems, are to changes in stimuli. Over time, what psychophysicists are trying to accomplish is to determine what the absolute threshold is to detect stimuli. And the absolute threshold isn't the minimum intensity to detect it, it's the intensity of about 50% correct that we can detect stimuli. So it's not 0 or 1%. I mean, 0 wouldn't be detecting at all, but it's not 10% for argument's sake. It's always 50. So you can see the S-shaped curve here, where the minimum amount of satisfactory ability to detect stimuli is set at 50%. And if you increase it beyond that threshold, you're more likely to detect the change in stimuli. Just noticeable differences are also an important characteristic of assessing the sensory systems. It's really just the minimal change in a stimulus that can be just barely detected. So if we talk about vision, and I mentioned a candle flame 30 miles away in a clear, dark night, how many more candles would have to be added before you can detect a change in brightness? Think about hearing and the dreaded ticking of a clock. If it's 20 feet away when it's quiet, how well can you detect this? And if we change the parameters, again, how sensitive is your hearing at determining or detecting the sounds in our environment? Touch, you can look at things like a fly's wing, which is falling on the cheek from 1 centimeter away, to see how sensitive you are detecting the wing on your on the skin. Smell, we use odorants, and a single drop of perfume can be diffused through an area equivalent to the volume of 6 rooms to determine how well we can detect these scents. And then with taste as well, we can assess this using things like sugar, as an example, dissolved in and get 2 gallons of water. Signal detection is relying on our sensory signals to perceive, or at least to send information sufficient that we can perceive them in the environment. But we detect these stimuli from whichever sense you want to think about through environmental noise. It's not like the world is perfectly quiet, if you will. When you try to look at stars at night and you're in a city, the noise or the pollution sometimes referred to is the other sources of light that are being emitted, making it harder to really appreciate and detect the number of stars in the night sky. Signal detection theory is a response to a stimulus that depends on a person's sensitivity to the stimulus in the presence of noise and on a person's decision criterion. So observers will consider the sensory evidence evoked by the stimulus and compare it with an internal decision criteria. Did you detect it or not? You can appreciate the challenges of detecting stimuli in the presence of other noise or competition. You've probably all been to a party, and in a party, everyone's talking, and think of all of those sound waves that are being emitted that reach your ear. And you have an uncanny ability that you can listen in on any conversation you want without even moving. You can filter out all of the other sounds, the noise, and focus only on the person you're talking to in front of you. Or conversely, you can eavesdrop on a conversation to your far left and filter out the person who's talking to you right in front of you. In signal detection, it's really about the sensitivity of our ability to detect these stimuli when they're present and in this presented in this what the source of other noise in the environment. In the real world, perception is an active process, and the resources enabling us to detect stimuli are limited. Many of us think we can multitask, but multitasking is very difficult. It involves, technically speaking, paying attention to more than one stimulus at a time. We like to think we're really good at multitasking, but the evidence does not support this. In fact, fMRI studies show decreases in brain areas during multitasking. So people who frequently multitask may often have trouble focusing on just one of them. These experiments show that using a cell phone while driving increases the likelihood of an accident by 4 times when you compare that to other modes of operating the vehicle, especially the control of nothing versus hands-free or text messaging. Frequent multitasking in academic settings can also have a negative impact on learning. I mean, think about you sitting in a classroom and the person in front of you has a computer open, but they're not taking notes. They're on Facebook. They're opening up other websites, looking at videos. Effectively, they're not paying attention to the class. Now, you might be thinking, well, how are they going to remember what's being said because they're not paying attention? But how is that also impacting your ability to pay attention? It's going to have a negative impact. Evidence suggests that even just in the presence of a laptop, it can detract from your ability to focus on the tasks at hand, consequently leading to a decrease in the ability to recall the information that was presented to you or to the group. So while we like to think we're really good at doing two things at once, the evidence just doesn't support that. Nevertheless, I understand the need to try to do multiple things at the same time. Fair enough. When we think of vision, we have to start first with the eyes to then further describe what the sensation to the perception of seeing. But you already know how we detect basic vision. You've all been to eye doctors, and effectively what they're trying to determine is your visual acuity at rest. How well can you see? If you rely on glasses to accommodate your vision or visual challenges, that's to ultimately give you the ability to perceive 20/20 vision as associated with the Snellen chart. So really, this is the smallest set of lines of letters that a typical person can read from a distance of about 20 feet. So if you have 20/20 vision, you're able to read the smallest line of letters that are presented to you at about 20 feet. Some people struggle to do this, and they would need corrective glasses to accommodate or to allow the light to bend a little bit differently in their eye to project on their retina to allow them to see the same thing. Now, personally, I was severely nearsighted, meaning I wouldn't see any letters, not even the biggest one at 20 feet. It would just be a blob of color. Of course, we'd have to use things like corrective lenses or glasses to correct that so that I would be able to see and detect what's there. Now, well, in a moment, we'll get into what the receptors are, but humans have not just receptors, you have millions of receptors in each eye that are selectively responding to certain wavelengths of light, of photons that are emitted by natural occurring sources of light like our sun, but also from artificial sources of light as well. The visible spectrum is 400 to 700 nanometers. So if you look at the bottom, you'll see there's gamma rays, X-rays, ultraviolet light. So these are shorter wavelengths, which you're not able to detect with your eyes. So we know that the wavelengths of light exist, but we don't have the ability to detect them. If you look further, you'll see the rainbow of colors from 400 to 700, which is then enlarged on the top. And this wavelengths that we can see present, that's what corresponds to the kinds of colors we can perceive, from indigo to green, yellows, orange, red. And if we look beyond 700 nanometers, there's infrared light, microwaves, radio waves. So these tend to be longer in their length, and we're still not able to see them. So it's kind of amazing when you think about all the light that you cannot see, which is the title of a very good book, by the way. But all we're able to actually detect is just that small wavelength between 4 and 700 nanometers. The properties of light that help determine the colors we see are based on their length, their intensity, or what we say their amplitude, and the purity of them. The length is just how frequently the wavelength is presented, that it occurs, if you will. The length is what determines the kind of color you perceive, the hue, as we call it. The amplitude or the height of the wavelength. So the length is from peak to peak, like the width of it, whereas the height is the amplitude, which determines its brightness, how bright something is. And you have lots of examples adjusting your cell phones accordingly in the evening or in the morning to make it less bright when or at least perceived to be less bright to us. We're not going to talk about how long it takes to adapt to bright or dimly lit conditions right now. And purity is just that saturation, the richness of the colors that we perceive in the world themselves. So, how do we see? It's important to note up front that none of this really is seeing, but this is the start of the perceptual process of vision. First, we have light. Typically, we talk about photons, but just as another reminder, sensation and perception is an entire course on its own that you can take if you'd like next year if you wanted. It's a 2,000 level course. But we first deal with light passing through the cornea. If you look at somebody else's eye, that's the translucent part that you see when you're looking to see the color of the iris that's behind the cornea. We see pupils, which adjust or accommodate the amount of light that can enter the eye, and then ultimately through the lens, which can then further adjust the light to be projected on the retina. Accommodation is the process by where the eye can maintain a clear, focused image on the retina, but specifically, it's the fovea in the retina. An important thing to note when we talk about color vision is that we're really talking about cones. So the way to help remember how we see color is to think the letter C for cones and C obviously for color. But in order to see things clearly, we almost have to forget about color at this point. If we want to look at something sharp, we stare at it, we move our eyes towards it. And that process is by projecting the image, the fovea in the back of your eye. It's an area that's large, only contains cones, which is why there's a large degree of visual acuity and color vision. The further you disperse from the fovea, there are still cones, but there's also more rods. And we'll talk more about rods and cones momentarily. But if the light that enters our cornea, passes through our pupil, is bent further by our lens, and ultimately projects not exactly on the retina, this is where we have difficulty seeing sharply. So if you are nearsighted, that just means the light that enters your eye is bent too hard, and it would converge to a point in front of the retina. So you won't be able to see things far, but you will be able to see things really close to you. So nearsighted is that you can see near, you can't see far. Conversely, hyperopia is where individuals that are farsighted have light that enters their eye, and it would converge. The light would come to a focal point behind the retina. Because of this, it often is the case that individuals that are farsighted can see off in the distance just fine, but they'd struggle to see things that are close to them. So if we look at the image here on the far left, it's the eye receiving light from, say, a coffee cup as an example. Now, the light, of course, reflects off of the cup, then passes into the cornea or through the cornea, through the pupil, the lens, and ultimately on the retina. And if that point, you see how it forms an X way out in front of the retina, that would be what we would typically call nearsightedness. Now, the image itself, we want the focal point to be back here on the fovea that you can see, but just for illustrative purposes, ignore the fact that it forms an X in the coffee cup. The image itself is on the fovea. We're going to assume that's normal. The image is kind of drawn counterintuitive, at least in my explanation, because it's not right. But let's just pretend if the coffee cup is projected and the image is right on the fovea, that's normal sighted vision. If we look at the image in the middle of column B, you'll see that the second image has the coffee cup in front of the retina. That would be nearsighted. And when the coffee cup, which is projecting behind the retina, which obviously it can't do, it's just showing that the light would converge beyond, which is why I don't like that they drew the X illustrating this. It makes it confusing. This would be farsighted. So what happens is that the retina is filled with a bunch of cells that can detect light. There rods, there's cones, then you have horizontal cells, amacrine cells, ganglion cells. The root ganglion cells form the optic nerve. Um, we don't necessarily need to get through all of the kinds of cells right now, but when light comes in, one of the important things to make note of is look what happens to the image. So vision's kind of weird in that the images we see in the real world are actually inverted or upside down when focused on our retina, but we see the world as right side up. What do you think would happen if we turned your eyeballs around so that the images in the world were presented right side up on your retina? If you guessed the world would be upside down, you would be correct. So the rods and the cones are really what we want to focus on as we talk about the cells that are responsible for our ability to detect light and ultimately the process of the perception of vision will take place. So the rods and cones are at the far back of the retina, or closer to the back of the eye, if you will, which means the light has to pass through the root ganglion cells, the bipolar cells, amacrine cells, horizontal cells. It just passes through all of these layers to then stimulate the rods and cones, which then project or send the information backwards or towards the inside of the eye, which will eventually exit out the retinal ganglion cells, forming the optic nerve. So the cones are for color vision, and they're shaped like cones, and the rods, they're shaped like a rod, and they're typically used to detect low light conditions, but they don't allow us to perceive color. They're really sensitive to low light conditions like at nighttime, whereas the cones are stimulated during the daytime. It's also important to look at our visual fields and how each eye has a left and a right visual field, and that each then sends specific information from the left and right eye. So we form binocular vision, and how through the optic chiasm, which is where the information forms an X or crosses in D, you can see behind the eyes. While we're not going to discuss things like the lateral geniculate nucleus, the superior colliculus, the information is effectively passing through. And then from the left visual field, it will go to the right hemisphere because of contralateral control. And from the right visual field, it will go to the left hemisphere. I know that's a lot to take in, but I would just probably pause here and review the images before you proceed. So the retina has two types of photoreceptor cells that I've mentioned, rods and cones. Cones detect color. They operate under normal daylight conditions, and they allow us to focus on very fine detail because they are exclusively in the fovea, only cones. So this is an area of your retina where vision is the sharpest. So when you turn your head and you look at something, you're staring at whatever's in front of you. It's sharp and detailed because you've allowed the light in your eyes, if you will, to focus your attention of that image on the fovea. Peripherally, or around the rod cones, are the rods, and they become active under low light conditions for night vision. Now, how good is our night vision? It's not bad. It's not great, but it's not bad. And you know that it takes only about a couple minutes to adjust to bright light. I'm sure in the morning you've grabbed your cell phone, turned it on, and your reaction's like, oh, that's really bright. But you'll notice after a couple of minutes, the perceived level of brightness has changed. Effectively, your eyes have adjusted or adapted to those conditions. But how long does it take for us to adjust to the night? You can think about when you go to bed and you turn the lights off. Your perception of your room might be it's pitch black. And if you've ever woken up in the evening, you'll notice that when you wake up, it's not nearly perceived as dark as it was when you went to bed. Now, it takes about 20 to 30 minutes for our eyes to adjust to low light conditions where we can more optimally see, if you will, the environment. But we don't see in color at night. We see in basically blacks and grays. You'll even find if you put images on the fovea, if you're trying to find the bathroom door in the dark, for example, or your bedroom door knob, if you look at where it would be, it disappears because the cones, remember, don't work in dimly lit conditions. It's the rods that do. So you'll look almost peripherally to navigate around the room, to feel for objects of where they are. The retina has layers of cells that also include bipolar cells, retinal ganglion cells. There's other, but we're just going to ignore it for now. But you also have a blind spot. And the retinal ganglion cells, that's what forms the optic nerve that exits the back of your eye. But we don't perceive a blind spot. But information that's presented there also isn't transmitted to our brain because there are no receptors there to detect it. So there is no mechanism for rods or cones to detect light because they're not present in the blind spot. The receptor cells in the retina form several layers. The innermost is where the rods and the cones exist. In the middle layer of cells, we have bipolar cells that convey neural messages from the rods and the cones to the retinal ganglion cells that are transmitting, if you will, the frequency of firing to determine what we're looking at eventually. And the outermost layer organizes these signals that sends them to our brain. The neural impulses travel along our optic nerve, and half of the axons code information in the right visual field, if you remember, and the other half code it for the left visual field. As the information exits the optic nerve, it travels to an area called the lateral geniculate nucleus, which is located in the thalamus. So the thalamus is a part of our brain that's a relay system where all sensory information passes through differently, though. They don't pass through the exact same area. They're passing through the thalamus, but different areas of the thalamus to other parts of our brain to be processed. This is true for all of the senses we're going to talk about today, except one, smell. Smell is the only one that does not go to the thalamus. It goes directly to the amygdala. But we'll wait later on to talk about this. The signals from the thalamus head to an area that we call V1. This is where information is first processed in your brain. Now, it's in the occipital lobe, which is the very back of your head. That's where vision begins. But it's important to note that in this area, it's not vision the way that you're thinking about it. Visual processing in area V1 is typically related to the processing of lines. Effectively, how we see the world, it's almost that it begins with building it up with just different line orientations. For example, if you look at the window and see a tree, you'll see there's vertical columns of the trunk. And then there's a whole bunch of branches that um, form after at the base or the top of the trunk. our ability to understand that it's a tree is based on our perception of the complete image, but V1 isn't the complete image. It's just breaking down the world into the road as a line that's perpendicular to a tree. And as we look again, it's not seeing, it's breaking and then rebuilding our visual world in the form of lines. Now, how do we know this? Hubel and Wiesel won a Nobel Prize for single cell recording in cats. And they did detect single neurons firing to specific line orientations. And there is more of that in this lecture. So I'll wait to save it for when it comes up. Now, how do we perceive color? Your cones, there's actually three of them, short, medium, and long wavelength cones are sensitive accordingly to each. So when we think about what is the perception of things like color, it's the processing of each of these three cones that are selectively sensitive to different wavelengths that our brain can interpret patterns of firing as colors. So the trichromatic color representation or theory of color vision is that patterns of responding across each of these three kinds of cones provides the unique neural code for each color. And if you're colorblind, then you likely lack the inability to perceive one of these wavelengths of light, resulting in your perception of color to be different. We also have color opponent systems that are pairs of visual neurons that work in opposition. So earlier I mentioned if you stare at a fixation point of a black and white image, or sometimes um, a field that has different colors than what we would expect to see in our environment. You stare at that for so long and then look up at the wall. In fact, you'll see an image appear on the wall, which isn't real. That's the illusion, but that's the other pair of neurons that work in oppositions to each other that produce this color after image. So when we see this image, which lasts for a few seconds, it's now in full color, what we would expect to have seen. So it's fascinating how we have come to learn how our eyes initially process light for our brains to interpret as to what they are in the real world. In class, I asked, how many colors do you perceive? And if we only have this short, medium, and long cones to detect these short, medium, and long wavelengths of light, you might get some creative answers. Is it three and just combinations of them? Is it 42 because that's how many crayons are on a box? Is it seven for the rainbow? But you can perceive millions of different colors of light. So we perceive a spectrum of colors. And it's all about how the light reflects off of objects in the world that transmit those wavelengths from the objects in the world to our eyes to then be perceived as the colors that they are. So color perception really is the sum activity of these three types of cones. Color mixing. This refers to the millions of shades of colors that humans can perceive that are products of light's wavelengths and how they mix ultimately the wavelengths from stimuli to absorb or reflect off of them in order for us, our eyes, to detect wavelengths of light, and then for our brain to process the neural codes that allow us to see color. Color deficiencies are a result because the three types of cones we have in the retina normally act as sources of information for our ability to perceive the world the way that we do. But there are genetic disorders that can result in things like being colorblind, where one of these cones is often missing. And in some very rare cases, two or all three are missing. These disorders are sex linked, not surprisingly affecting men more than women. Because if you're a woman, you have two X chromosomes for each cell, which is allowing the likelihood that if one parent contributes color blindness and the other one contributes the other X chromosome and it doesn't have it, that the dominant trait will win out, meaning you won't be colorblind. But for a man, if you inherit the X chromosome from your mother and the color blindness is then passed down, and if your father doesn't have an X, obviously contribute because it's the Y, then we're left with you're colorblind. Color opponent systems can explain color after effects, which I mentioned just briefly a moment ago. That's when these pairs of cones or these channels, they work in opposition to each other, perceiving an image in the environment that we will that isn't real. That's the image we see that's projected. We simply look at a wall. It's as a result of the opposite systems sending signals to our brain that this is what was active, but the opposite signal of it. So area V1 is where light from our eye to our retina to the optic nerve through the thalamus, other structures, we'll just forget that, is sent to be processed. And this is a part of the occipital lobe that contains primary visual cortex. Particularly, this area is sensitive to edges, different feature detectors that respond to very particular orientations of objects in space. And all of these edge detectors really work to distinguish objects in the environment from each other, the lines that I just described a moment ago. We know this is true because animal research, particularly was in cats, but here they're showing you you know, a primate species. And what is critically important in our understanding of how these neurons fire is that if you look at the first orientation in the top, you'll see that 45 degrees is the preferred orientation for this particular neuron to fire, meaning it fires the most when the line is at 45 degrees. But if we record that same neuron and we just make it vertically 180 degrees for argument's sake, if we rotate it to the left 45, in this instance, it still fires, but it fires less frequently. So they have their preferred orientation, but it's not fixed that they don't fire if it's not exactly it. It just fires less frequently. And the further we get away from the preferred orientation, the less recordings that will be observed from that particular neuron. So the main piece to understand is that they have their preferred orientations to fire to, and they will fire in the presence of similar orientations, but they will fire less frequently. So from V1, effectively the information very quickly gets sent to other areas of our brain. We're really going to skip the other areas, V2, there's 3, 4, 5 as well. But now what's important is to think about what is in our environment and where it's located. So the visual system ends up forming different pathways, one from the occipital lobe and the other through the ventral lobe. The ventral stream is the what things are. Think of our memory of knowing what objects are in the environment. And if we know what they are, we may be asked, where are they if they're presented in front of us? So what is an object? What do we do with it? What is it used for? And where is it? Now we know these are distinct, separate pathways because, unfortunately, some people have suffered brain damage here. And as a result, these streams have resulted in the inability to necessarily know what something is, but you could detect where it is. Meaning, you could pick up a brush, but you wouldn't know what to do with it. Conversely, you might know what a brush is used for, and you might mimic brushing your hair, but if one was on a table with other objects in front of you, you wouldn't know where it is. So these forms of visual agnosia, the inability to know what or where something is, have been determined because people have suffered brain damage where that's exactly what happens with them. The ventral, the what stream versus the where it is, the dorsal, the top part of your head, the parietal lobe. Individuals were unable to determine the opposite of what they could do. So if they knew what an object was, they wouldn't be able to pick it up versus they would know where it is. They could pick it up, but they wouldn't know what to do with it. So effectively, it's a binding problem. Now, the binding problem really is how does the brain that takes in all of the sensory information create a visual world that has combined all of this information smoothly, accurately, to allow us to see the world the way that we do? We don't really know how our brain does that, but we do know how it gets all of this information. So we know a great deal about what it can process. We really just don't know how it puts it all together. What do you think? So effectively, the binding problem is how features that are linked together by the brain do so in a unified manner that allows our visual world to be perceived as together, united, and not free floating or in combined or uncombined parts. Largely, we know that our brain processes things in parallel, which allows us to perform multiple activities at the same time. Now, while we can't necessarily focus our attention on multiple things, not very well, our brain's really good at doing this, effectively to a different degree, though. It's able to take in all sorts of information and effortlessly combine them to allow us to see the world and experience the environments we're in as we do. The binding that we talk about can also lead to perceptual mistakes, a whole bunch of different illusions. So, an illusory conjunction is a perceptual mistake where features from multiple objects are incorrectly combined. So, really, when we think about how we come to know about the world, it's more think of feature integration theory, which is when we focus our attention, it's not required to detect individual features that compose a stimulus, but our attention is required to bind them together, which allow us to perform visual searches and to pay attention to objects that we see. So when we think about images that are presented here, this is illusory conjunctions. And this occurs when features such as color and shape are combined incorrectly. So if you look at A, the task would be to remember what the color of A was and the letter X as an example. So the correct, what would be reported would be A, red, X, blue. And sometimes the illusion is that it's the opposite colors reported, would say A is blue and X is red. So they got the letters correct, it's just the colors were backwards. Sometimes people incorrectly remember a letter. So the A is red and it's correct, and it was a blue T. So the color was correct, but the object itself wasn't. And sometimes there's misreported colors as well. So they say a completely different color was present when it wasn't. But this is pretty rare. It's just demonstrating that it can happen and that the results that we see when we ask people to report what was presented to them really isn't a result of just guessing. There is an intention, strategy behind this, or the detection of features. The features themselves become integrated, which is the name of the theory, where that our attention, I said, is not required to detect the individual features that make up a stimulus, but it is required to bind them together. How do we perceive the world? How does our brain organize events? One of these views was argued to be a modular perspective, that our brain has highly developed specialized areas that are dedicated to very specific processing of stimuli that our brain receives from the senses. Meaning, you have an area called the fusiform gyrus or the fusiform face area, and its role, largely, there's a couple other exceptions, is to detect faces. So the fusiform face area is literally an area that detects faces. And if there's damage that occurs here, it's not surprising, you would guess, you would never see a face again. And that's exactly what happens. So that's a modular perspective in that this area, the fusiform gyrus, the bottom of your temporal lobe, is highly specialized just to detect or allow us to perceive faces. Different regions of the ventral stream respond preferentially to different kinds of objects, such as faces, bodies, scenes, tools. And we do prefer to look at faces, just as infants prefer to look at faces. The distributed representation of object categories is really, there's different patterns of activity across multiple brain regions that allow our brains to code and identify what these objects are. So remember, we're all using our eyeballs, if you will, to focus our attention on objects, which first are broken down by their basic features, line orientations. Ultimately, the patterns of firings from, we'll say largely our cones at this point, how each of the short, medium, and long wavelengths fire uniquely to objects. The coding of that unique object gets remembered as things like horses, hammers, whatever you want to think about, faces, different features of faces, and how that forms identities of the friends we've had or our family members. So there's a lot that goes on, really. And I know simply how do we see? We open our eyes and there's the world. But simplistically, this is more or less what's going on. Maybe there should be a significant emphasis on simplistically. How do we recognize objects then by sight? So we have to form memories of what the things are. And another perspective on this issue examines how these neurons, in particular, how single neurons in our brain can respond to objects as well as faces. Now, it's important to understand we have perceptual constancy, which is that perceptual principle that states even as the aspects of sensory signals change, our perception of things remains constant. We also have conceptual knowledge of the world. So when we perceive an object, we don't merely recognize what it looks like, but we understand what it is, the characteristics of it, and the significance of these things to our own behavior. The principles then of perceptual organization is really a process of grouping and segregating features in our environments to create whole objects that are organized in meaningful ways. So imagine you're me standing in the classroom looking at students. What's uniquely in front of all of students are desks. And the desks, if you will, separate your lower torso from your upper torso. It's not that when we see this, we assume there's two parts of you now. We still know you're a singular whole. So that perceptual organization that we know people are still a whole unit, not separated just because there's a table in front of you, is a result of something we call Gestalt psychology or grouping, that you are the whole and not just the sum of your parts. So the grouping allows us to perceive things and organize our perception of the world based on simplicity, how objects could close, how they continue. Like think of railroad tracks, continuity going off of parallel lines in the same direction, how similar things are. Think about when you see people that are in close proximity. We must assume they're all together, they're part of a group, as well as common fate. So simplicity implies something like a direction. You can see how in B there's no closure. The lines aren't complete like they are in A, but we still think that that's an arrow pointed upwards. In C, we can imagine each of these lines as string and how that there's two pieces that just intersect, but they continue on their own path. If it's similar, we assume all the triangles, the circles, and triangles go together. Just like proximity, we assume there's three groups. And common fate is just the perception of things moving towards the same direction, experiencing the same things. Gestalt principle at work is that above, when we look at some images, it might look meaningless to us, meaning it's hard to really detect exactly what the whole thing is, but we can see some of the pieces of it. But down below, if we turn that white set of lines to black, now it's a lot easier to see that it's an airplane. How or why, do we think these things occur? It's pretty fascinating when we think about this. But if we don't have a whole lot of experience looking at images and trying to detect what they are, you can see how it might be more difficult to do so. But with practice, of course, you learn how this works. You're effectively training your perceptual sense system to use the sensory information to gain knowledge about what it is. And now you'll effortlessly and automatically start seeing what the scene actually is. It's also important that we discuss separating the figure from the ground. And you've all seen, I'm sure, the Rubens vase illusion before, where if we look at the white as the foreground and the black as the background, it looks like a chalice. But if you see the white as the background and the black as the foreground, it looks like you can see two faces that are staring at each other. So the forehead, the nose, and the mouth, the chin, and it's mirrored on the opposite side. So figure ground is about what we look at that we think is in front of us versus what we perceive to be in then behind us. How we perceive things like depth and the size of objects depends on monocular depth cues. So these scenes in our environments are projecting on our eyes. And we have lots of information about the depth of field, especially when we view them with one eye, the relative size, how familiar they are. We have linear perspective. We get texture information, position, the relative height of objects. Binocular cues really is disparity or binocular disparity is the difference in the retinal images of the two eyes that provide information about things like depth. And we know we can create scenes that can trick our brain into perceiving objects as larger than what they are. I mean, take this as a simple example. If we manipulate the location of a person in a scene, so on the left, that looks pretty typical of what we would expect, meaning the person that fills up more of our retinal image is perceived to be in front of us, and the person in the back that um, perceives a smaller retinal image is perceived to be off in the distance down the hall. But if we take that same person in the end of the hall, we move them up front, what happens? It's no longer perceived as, say, normal because that doesn't make sense. Visually, this doesn't make sense. Whereas on the left, it does, but on the right, it wouldn't. So the idea that it's further away is lost. And it's like, what's going on here? It's harder to really understand why this would happen, even though the image is exactly the same. You've all, I'm sure, stood over railway tracks to see how they move together in parallel, often towards the end. You can look at other things like texture and shape, like the sand of a desert that's dried out, no different than depth of the mountains and the flowers in the front, um, or just the interposition of the fruit laid across a table as a display. And the majority of us are perceiving the world, of course, with two eyes. We have this binocular disparity where the world is perceived in three dimensions because our eyes are a distance apart. So the images that we look at, they fall slightly different in the location of where they are on the retina of each eye, just slightly so. So in this scenario, the images of the square and the circle fall in different points of the retina of each eye. And the disparity in the positions of these images then provides a compelling cue in how we perceive things like depth. And we can also manipulate perception of depth and based or changing the size of the person, but the depth is the illusion of changing or engineering a room that really, on the one hand, we perceive to be a square room when the reality is it's not, it's a rhombus. And by changing the actual shape of the room, we can trick our brain into perceiving the objects in it as very, very different than what they are, which is why some people are perceived to be quite tall and others to be quite small because of how close they are and how much information is then perceived on the retina from each eye, from each location. Not only can we perceive obviously things like color, size, and how we group the world together, we also perceive things in motion. And to sense motion, how things are moving, the visual system must encode information about both time and space. And the medial temporal lobe in the dorsal stream is what allows us to perceive these time and space positions of things to allow us to see motion. We can also see waterfall illusions, which is analogous to color after effects. So things appear to be moving after the fact, as well as apparent motion. I'm sure you've all parked your car in a parking lot. You turn your head to the left or right to see a vehicle next to you slowly backing out, and you panic thinking you're moving when it's actually them. So it's basically the perception of movement as a result of alternating signals that appears in rapid succession from different locations, creating the illusion that we're the ones moving when we're not. Perception of motion of other people is, we know that we are in motion. It's not uncommon then to think about biological motion perceptions that we're able to perceive things that are moving as really identifying individuals or socially relevant features of people in society. But think of how critically important it is to just to detect motion. Think if you're in the park, you're in the woods. Obviously, we know if the trees are moving, it's due to the wind. But if we ask kids why they move, they might think, well, the tree's alive, so it's moving. But we're also really good at detecting motion in the woods. And if we do, what is it? What's the object that we've detected? This is where features of the objects are important because is it just a squirrel? Was it a bear? And what other features would go with seeing? Of course, we think things like hearing as well, but we're not quite there yet, are we? We also can talk about sensory superpowers. And, you know, this is pretty fascinating as well, is that every domain has a superpower. And the visual system itself has an amazing spatial acuity. It's really, our eyes, our visual system is really good at distinguishing between two features that are very close together in space. The auditory system has temporal acuity. So we're really good at distinguishing the time that sounds reach our ears, even if they're very, very close in time. And we can capitalize on these strengths in everyday life. So think of ventriloquist illusions. How many of us can be not fooled, but amazed that someone can project their voice without moving their lips and say all sorts of complex speech as if it was attributed to things like a puppet? It's quite remarkable. It's even remarkable when we think about the visual system is so, if you will, super that it can override the ability of our ears to process the correct signals they receive. So if you Google the McGurk effect, you can see how even if we hear a constant sound played, because of what we watch, of how the lips or the articulators are modified to look like they're producing different sound, our perception of it will be different. It's quite remarkable. Maybe I'll send a link of that so you can watch to see. Because it's surprising if you think about the ability of vision to supersede another sense, which it will. And the cool thing is, even if you know, it doesn't matter. The McGurk effect is such a powerful, um, well, for lack of a better word, effect that it will work whether you know what's happening or not. It's also fun to think about other challenges to perception. One of these is change blindness, or really inattentional blindness. Imagine a scenario you're walking on campus and someone stops you and says, can you tell me where the library is? And they hand you a map. Well, people have done something like these many times where they just stop students, give them the map, and say, where's the library? And then very quickly, you replace the person who's asking for the directions with somebody else. Somebody brand new steps in. The study is not to find out where the library is, it's to determine whether you can detect a whole different person asking you for directions. And you might think, oh, no, there's no way that would happen, but it does. Even though we see the world, we're not always great at detecting changes in it. Now, some of us obviously are better than others. But in these kinds of instances, you'd be surprised how many people fail to detect a different person standing in the door. Another fascinating ability of vision is synesthesia. In my experience, when I've taught intro psych, um, many students aren't aware that they have synesthesia until university. Now, what is it? It's the perceptual ability to perceive something like color with sound, for example, or you see numbers as different colors, and they can also have different personalities. So it's one attribute of a stimulus, such as sound or shape, leads to the conscious experience of additional attributes, often in different sensory modalities than the initiating stimulus. This is differing for different subjective experiences of synesthetes and non-synesthetes, and it can produce striking differences in how they perform basic tasks. So imagine you're trying to do math and you have visual perception of color with number. it can actually benefit them in doing math computations. And the reason that no one really knows that they have this is it wouldn't affect you, um, in a sense that it's not going to make you worse at doing things. It might benefit you. One of my friends, her entire doctoral dissertation was looking at synesthesia and how they perceive different stimuli in the environment. So scientists are trying to understand how the brain may differ in synesthetes than non-synesthetes. One of the theories is that everyone possesses the ability to do this in early childhood, but as our brain develops, the senses delineate from each other. Whereas in synesthetes, they don't. They somehow stay a little bit fused. But the results to date are conflicting. There is no real evidence for a genetic connection here. But nevertheless, it is fascinating that this exists, and it might be you. I think in class, I have one person that had right now the ability to perceive color when sounds are played. I think what I'll do is break the lecture up from here because there's still quite a bit left, and I think I've talked long enough for now. So we'll take a break here. We'll come back in part two, and it'll be hearing and the remaining senses. I hope you've enjoyed the lecture so far, and we'll speak to you soon. Thanks. Bye for now.